My name is Michael Mackey. I'm speaking to you from my study here in Eastern Canada in Montreal. And today's lecture is going to try to trace a little bit of the interactions between the biological sciences, mathematics, and physics over the last few hundred years. I'm not going to look at the role of physics and engineering in the development of diagnostic tools in medicine, but rather I want to look at the way in which the physical and mathematical sciences have actually influenced the development of the biological sciences. So if we look back into the 1400s with Copernicus, the 1500s with Galileo. So Copernicus was the one, of course, who came up with the notion that the sun was actually the center of our solar system, and which, of course, at the time was quite heretical. Galileo, 100 years later, really promoted this idea with his observations. Then 100 years after that, the Italian Galvani was interested in studying so-called animal electricity, while Thomas Young, who first did the two-slit experiment and demonstrated the wave nature of light, Leon Fouquet, who developed the Fouquet pendulum and related the behavior of that pendulum to the rotation of the Earth, and then Hermann von Helmholtz, who we'll get to in a bit, was a very famous physicist in the 1800s. But interestingly, all of these people who we consider to be physicists today, in point of fact, were trained as physicians. And this is not surprising because in the educational climate of their times, the only three things that you could really study were law, theology, and medicine. So if we go ahead a little bit later to the era of the American Revolution, the 1700s, Benjamin Franklin was a revolutionary philosopher and also an amateur scientist. And he was interested in what exactly static electricity was. And so Franklin did these somewhat bizarre experiments when he would fly kites in thunderstorms, and hoping that the kite would be hit by lightning so he could charge up a Leyden jar, which was a primitive way of storing electricity. And then a different type of electricity attracted the attention of people like Galvani and Volta. So Galvani had observed that during thunder and lightning storms, he would see twitching in frog carcasses. Don't ask me how this came about. I have no clue. But he would do rather bizarre experiments when he would hang freshly dissected frog legs on a metal fence outside his villa during a thunderstorm and observe when and how the frog legs would twitch relative to a thunderbolt. So Vito Volta, not Volterra, but Volta, was also interested in animal electricity, and he had a counterintuitive, or not counterintuitive, but counter explanation that was opposed to the one that Galvani came up with. And Volta's actually explanation is we now know is the correct one. So if we go ahead 100 years into the late 1800s, there was a very famous German physicist by the name of Hermann von Helmholtz. Helmholtz, as I said earlier, was actually trained as a physician. And the story is an interesting one because Helmholtz's family, his father was the headmaster of a gymnasium in Prussia. And the family, although relatively well off for the time, certainly did not have the money to send Helmholtz to university. So Helmholtz did what many young people do, even today. He joined the army, the Prussian army. The Prussian army sent him to medical school. He studied medicine, graduated, and then spent 8 years working as a physician within the Prussian army. And after that, he took a teaching position in Berlin. And in the late 1800s, he wrote a rather incredible 8-volume treatise on, it was entitled Lectures in Theoretical Physics. And this covered everything, pretty much everything that was known at the time, namely celestial mechanics, electricity and magnetism, kinetic theory of gases, such as it was known then, statistical mechanics, thermodynamics. And Helmholtz was an incredible polymath. So he not only was well-trained, personally well-trained in physics and mathematics, but he also worked with other surgeons and physiologists. He co-wrote or co-authored a book on the anatomy and function of the inner ear of humans. He also wrote on his own a so-called Handbook of Physiological Optics, which is even still in print today. He was the first person to measure the conduction velocity of the action potential. He measured the amount of heat released when the action potential took place in a nerve. He developed the ophthalmoscope and the stethoscope, or refined the stethoscope. So he was a rather amazing person. And the fact that he was so amazing is actually celebrated by his university in Berlin, Humboldt University, with this rather marvelous statue that's out in front of it. If we go ahead just a couple of decades after Helmholtz, A.V. Hill was a student of mathematics at Cambridge. He was reputedly quite brilliant, but somehow was enticed to study physiology while at Cambridge. And so after essentially finishing his entire mathematics education, he switched to physiology and became one of the leading British physiologists of the century. He was very interested in the nature of nerve excitability and developed the so-called chemical wave theory, which turned out to be wrong. But he published this in a book in 1932. And after that publication, a young German Jew by the name of Bernard Katz escaped Berlin. He had done his undergraduate work in Berlin. And he came to the UK in 1933, where he studied under A.V. Hill. And not only did physiology, but also studied mathematics and physics. Now, Bernard Katz was a brilliant scientist who contributed much to single-cell physiology. And perhaps the most noteworthy thing that he did was to develop the concept of quantal transmission, quantal synaptic transmission, with his graduate student, Paul Fatt. So the Fatt and Katz paper was a marvelous example of experimental design, data collection, analysis of the data within the context of a mathematical model for the transmission properties at the neuromuscular synapse. And I think it's the first paper that I have ever seen in which the word chaos was actually used. So Fan and Katz developed the concept using a stochastic theory of quantal transmission at the synapse. And this earned them the Nobel Prize in physiology and medicine. Now, if we just back up a little bit to the late 1800s, Walter Nernst was a physical chemist in Berlin and was extremely interested in the transport of electricity and electrolytes under the action of either an electric field or a concentration gradient. And in conjunction with Max Planck, a physicist who's better known for his development of the concept of the light quantum, a theory was developed which is now formulated within the context of what is known as the Nernst-Planck equation. And the Nernst-Planck equation describes movement of ions under the action of a potential gradient or a concentration gradient. Now, Nernst himself was brilliant, as well as was Max Planck. Planck, of course, is renowned for his contributions in the field of quantum mechanics, quantum physics. And like Helmholtz, is honored by a plaque and by a statue in front of Humboldt University. Humboldt University had a lot of very famous people. Now, there's also a very interesting Max Planck Institute for the History of Science in Dahlem. Dahlem is a suburb of Berlin, where many of the Max Planck Institutes are located, and the Max Planck Institute for the History of Science is also located in Dahlem. And they've done actually wonderful research documenting the development of science, both physical sciences as well as biological sciences. But to go back to the Nernst-Planck equation, an American physicist by the name of Kenneth Cole was at Columbia University. Like most people who grew up in North America as academics in the 20s and 30s, was able to read German and read the Nernst-Planck papers and realized that the Nernst-Planck equation was precisely what he needed to describe the movement of ions across an excitable cell membrane. So he used the Nernst-Planck equation, the development that went with it, to actually study the behavior of the impedance of a single cell from the squid giant axon during excitability. So here you see this solid line right here, the outside line, which was then obscured here, and there's the end of it. That is the action potential in a squid giant axon. So this is about 70 millivolts from the baseline up to the top. And from here to this end is about 2 milliseconds. So it's a very short, explosive kind of behavior that you see when the action potential is generated. And what Cole and Curtis did was to demonstrate that during the action potential, there was a huge increase in the conductance of the membrane, which is shown here by this wide band that gets bigger at the maximum and then tails down. Now, Cole's work was extensive, and it went on throughout World War II. And before and after World War II, he had been visited by a couple of British physiologists and mathematicians, Hodgkin and Huxley. They both were well trained in physiology as well as mathematics, having come from Cambridge. And they realized that they had at their disposal the techniques for actually figuring out what ionic currents were giving rise to the action potential in squid. And so they had a series of 4 experimental papers in the Journal of Physiology in the late 40s and 1951, in which they carried out a beautiful set of experiments, really looking at the behavior of the movement of sodium and potassium across the membrane as a function of time, as a function of potential. And then in the final paper, they developed a complete theory based on their experiments and data in the first 4 papers. And in this final paper, they were actually able to quantify all of their data into 3 nonlinear differential equations and 1 partial differential equation that described the propagation of a traveling wave dependent upon the 3 other equations. It was actually an amazing feat, particularly for 1952, and very rightly earned them the Nobel Prize in Physiology and Medicine. Now, how did they do it? How did they solve this 3-dimensional system of nonlinear ODEs in combination with this PDE that described the propagation of this traveling wave? They did it using a bank of hand calculators like this. This is an example of one that's on display in the computing laboratory of Oxford University. And they had a bank of women essentially carrying out a series of steps. And it took them 1 working week, that is to say, 5 days of 12 hours per day to solve these equations to actually give the propagated action potential for a period of 2 milliseconds. That's dedication for you. So Hodgkin and Huxley, a pair of brilliant physiologists, brilliant mathematicians. And Huxley was no slouch. He went on to actually look at the behavior of muscle contraction. So here you see Huxley in his laboratory about 1954. In 1954, with his postdoc Niedergerker, they really looked at some of the structural changes in striated muscle during contraction. And later that same year, Huxley published a monumental review paper, which not only was a review paper, but it also laid out the cross-bridge theory for muscle contraction. So it was based on the physics and developed the mathematics of how actin and myosin interacted with each other to actually produce muscular contraction in striated muscle. Now, one of the interesting things about the Hodgkin-Huxley equations is that they have so-called fast and slow variables in them. It's a 3-dimensional system. The ODEs are 3-dimensional. And a young physiologist, mathematician named Richard Fitzhugh was interested in these. And he thought there might be a way to actually simplify our understanding of what was going on with the movement of sodium and potassium by looking at a simplification of these equations. Now, Fitzhugh himself was an interesting person. He had been a student at UCLA, Los Angeles in 1940, 1941. And his family was relatively privileged in the sense that they owned a few ships that sailed in the Pacific, and they offered cruises to passengers. However, in December of 1941, Japanese attacked Pearl Harbor, and then the Americans declared war upon Japan. And as you can imagine, having a cruise ship company in the Pacific with a war on Japan going on probably wasn't a very good idea. And in point of fact, the US Navy commandeered all the Fitzhugh ships as using them for troop carriers. So Fitzhugh no longer had the money to continue in university, so he worked as a draftsman at the Boeing Airplane Company in Wichita, Kansas, throughout the war. And after the war, he went back to the University of Denver to complete his degree in physics and mathematics. While he was there, of course, he had to demonstrate proficiency in a foreign language. In those days, we all had to actually demonstrate that we could either speak and read another language like French or German or Russian, and Fitzhugh chose Russian. Very interesting and very, very timely choice, because when he eventually started looking at the Hodgkin Huxley equations, he realized that these fast and slow variables in the terminology of the Russian dynamical systems literature could actually be approximated. And he came up with an approximation, which is now known as the Fitzhugh-Nagumo equations, in which he reduced this four-dimensional system to a two-dimensional system. And by doing that, he was able to gain great insight into how the action potential actually occurred. And the Fitzhugh-Nagumo equations are still studied today and still of great use for neurophysiologists. Now, this is a picture of Fitzhugh. When I wrote to him back in the 1990s, I was interested in the story, and he was very kind and wrote back to me and gave me a great deal of the background and history for this whole scenario. Now, it had been suspected for many years that the way the sodium and potassium ions moved across the membrane of an excitable cell was through tiny little channels. You know, one notion might have been that the ions would just diffuse through the membrane. But another concept was that in point of fact, the ions were moving through specialized holes or pores in the membrane. But it was very difficult to actually prove this experimentally. And then two scientists, one of them, Erwin Neher, right here, a German physicist, theoretical physicist, teamed up with a Bert Sackman, who was an American no, I'm sorry. Anyway, Neher and Sackman teamed up and developed a so-called patch clamp technique for actually measuring the current flowing through a single channel in the membrane. And you have to realize these channels are on the order of 4 to 5 angstroms in diameter. And there are maybe 30 or 40 per square micron. So you have to actually have a very fine tipped electrode that goes down, touches the membrane, you suck up a bit of the membrane, and you hope that you've managed to isolate a channel and you're recording from it. And Aaron Sackman got the Nobel Prize in Physiology and Medicine for developing this technique and developing the analysis that allows people to actually look at single channel currents these days. And this is an extremely important area, particularly in pharmacology, because the technique is used to actually assess the efficacy of drugs in terms of, for example, cardiac function and things like that. So this whole business of looking at single cells is related in a way to actually looking at the behavior of nervous systems, where you have collections of nerve cells communicating with each other. And one of the most interesting papers is that of a 1943 paper by Warren McCulloch and Walter Pitts that was published in the Bulletin of Mathematical Biophysics. And it was called The Nervous System, and it was called A Logical Calculus of Ideas Immanent in Nervous Activity. And Pitts and McCulloch basically laid out all of the possible logical combinations that you could produce with elements that were 0, 1. They were either on or off. And so this is essentially at the foundation of modern computing, and it's also at the foundation of modern artificial intelligence and machine learning. It's a wonderful paper, totally amazing. Another extremely prolific mathematician who was interested in the biological science was Norbert Wiener. Now, Wiener, we know, contributed a great deal to theory of stochastic processes, to quantum mechanics, but what is little known is that he was also extremely interested in problems in biology, and he worked with a Mexican cardiologist named Arturo Rosenblad for years. And that interaction with Rosenblad very much influenced Wiener's thinking, and he was then led to develop the concept of the field of cybernetics, and he wrote a book called Cybernetics, or Controlling Communication in the Animal and the Machine, in which he likened physiological systems, living systems, to basically machines. Now, this was a rather astonishing notion at the time in the early 1960s. Another person looking at simple nervous systems was a man named H.K. Hardline, who was at Johns Hopkins University for years. He studied at Johns Hopkins. He studied physiology. After he received his PhD, he got money from the National Science Foundation to spend 2 years in Europe, and he spent 1 year in Leipzig with Werner Heisenberg, and a second year in Munich with Arthur Sommerfeld, because he wanted to learn theoretical physics. After his stint in Europe with Heisenberg and Sommerfeld, he went back to Johns Hopkins and worked for his entire life studying the simple, relatively simple nervous system that is in the eye of the horseshoe crab. And there's a wonderful collection of papers, a mixture of experimental and theoretical papers in the book on studies on excitation and inhibition in the retina that are basically all from Radloff's laboratory. And they're all marvelous examples of meticulously designed experiments combined with data analysis and simple but appropriate theoretical modeling. Now, the interesting thing is that Hardline was also the thesis advisor for Fitzhugh. Remember Fitzhugh from the Fitzhugh-Nagumo equations? Well, after he left the University of Denver following World War II, he came to Hardline's laboratory in at Johns Hopkins, and he received his PhD there. And from there, he essentially moved from Baltimore to Bethesda, and he spent his entire professional life in Bethesda at the National Institutes of Health. Alan Turing. We're all familiar with the sad story of Alan Turing and the shabby treatment that he received from the British establishment. But Turing himself was a major contributor to the development of our concepts in the biological sciences and in neuroscience because of his understanding of the McCulloch-Pitts modeling that was published in 1943, his application of that to machine learning, and which he also applied in terms to his efforts at code breaking during World War II at Bletchley Park. Another pioneer in this area who's little known is a physicist by the name of Frank Rosenblad. And Rosenblad was very intrigued with the possibility of artificial intelligence, and he built on the concepts of Turing and McCulloch and Pitts, and he built a prototypic neural network. This was not one that was built in a digital computer, but it was actually an analog computer. And it was called the perceptron, and it was able to do simple calculations as well as perform simple memory tasks. So just at the end, I'd like to switch. We have spent a lot of time talking about developments in the neurosciences, but I want to turn to a different area, so-called the area of molecular biology, because it's extremely timely. It's a term that didn't exist 50 or 75 years ago. And it really had its start with these two people, in my opinion. This right here is Niels Bohr. As you may remember, Niels Bohr was the author of the so-called Copenhagen interpretation of quantum mechanics. And he was also the father of the concept or the so-called complementarity principle in quantum mechanics. And this man right here, this younger man, is Max Delbruck. Now, Max Delbruck received his doctorate in 1931, I think, and he went to spend a couple of years in Bohr's group in Copenhagen. And then Delbruck came back to Berlin in late 1932 or early 1933. It was a bad time to be going back. And he came as an assistant to this woman here, Lise Meitner. Lise Meitner was an equal co-worker with Otto Hahn in one of the German institutes in Berlin. Hahn is well known, got the Nobel Prize in 1944 for his work on nuclear fission. It was actually the work of Lise Meitner on nuclear fission. And, Meitner was essentially forgotten for many years, but is now recognized. And she died when she was 90 years old, I'm sorry. And there's a very nice plaque here on the outside of her workplace in Berlin, recognizing her contributions. And there's a wonderful biography by Ruth Syme about Lise Meitner, which, if you like scientific biographies, and I do, I think you would find it very, very rewarding to read. Anyway, Delbruck shows up in Berlin, 1933. Bad time, really bad time. And of course, the Nazis are in power. They are choking off all kinds of meetings. If you want to have a meeting with more than 3 people, you have to have a party member present. And so Delbruck and some of his younger colleagues in the Institute had an evening seminar that they ran in different people's apartments. And as a consequence of that seminar, there was an incredibly influential paper that ended up being published in 1935. And the German title translates Concerning the Nature of Gene Mutation and Gene Structure. Now, this paper was virtually unknown for years. But what happened in this paper was first of all, they had experimental data where they looked at the effects of gamma rays and X-rays on Drosophila and mutations in Drosophila. And then they analyzed these results theoretically, the data. And then Max Delbruck, put forward a model, a theoretical model for genetic mutation based on atomic physics. And their conclusion was that a mutation was a molecular rearrangement within a particular molecule, and the gene was simply a union of atoms in which a mutation can occur. Well, that doesn't sound particularly earth-shaking in 2022, but I can tell you in 1935, it was earth-shaking. That paper was essentially forgotten for years until it was picked up and recognized again by Erwin Schrödinger. Schrödinger, of course, the father of the Schrödinger equation, was also in Berlin in 1932 and was so disgusted with the success of the National Socialist Party in attaining power that he wrote a resignation letter and was not circumspect in his criticism of Adolf Hitler. And he took off with his family to Vienna, where they set up house, and they thought that they would be safe there. But of course, the Germans annexed Austria. And so things again became quite uncertain. Schrödinger and his family escaped Austria just in time, just before the annexation by the Germans. They went initially to Oxford. He tried to get a position at the Institute for Advanced Study in Princeton, New Jersey, and then eventually ended up in Dublin, Ireland, at the Institute for Advanced Study. Now, Schrödinger had a pretty unusual personal life, relatively unusual. He was married. He and his wife had a child. She had an affair with a colleague of his and had another child with that colleague. And they lived with a second woman who was described in official documents as an au pair girl. That is to say, somebody to take care of the children, but in point of fact, she was Schrödinger's mistress. So we have Schrödinger's wife, and Schrödinger's mistress forming a family with several children. He didn't get the position at the Institute for Advanced Study because they offered him half the amount of money that Einstein was being paid. Schrödinger was so irritated by this that he turned down their offer. But it now appears that based on records that have been uncovered, they offered him this small salary because they really didn't want him to come. They were afraid that his unusual family arrangements would offend people living in Princeton. Anyway, if you want another interesting biography, try Schrödinger's Life and Thought by Walter Moore. Excellent book. So in Dublin, Schrödinger had only one duty. Well, two, actually. Number one, he had to think And number two, every year he had to give a series of lectures to the public. In 1943, he gave a lecture series that ended up being called What is Life? And it was transcribed, no doubt, by some hapless graduate student and eventually published in 1944 by Cambridge University Press. Still in print. And it's still worth reading. You can read it in a couple of evenings. It's very, very good. A lot of it's inaccurate, but a lot of it is dead on. Very interesting. In that book, Schrodinger was quite taken with the results of the Delbruck paper from 1935 and talks about them at great length. And it's clear that the Delbruck results with Delbruck's German colleagues in the early to mid 30s greatly influenced Schrodinger's thinking. This book was extremely influential. After World War II, of course, the book was recently published. There were a whole host of young theoretical physicists that had been working in the war effort, British as well as American. And they were extremely disillusioned by the outcome of the war and the destruction of Hiroshima and Nagasaki by the Americans with the 2 atomic bombs. 7 of these people, Seymour Benzer, Sidney Brenner, Crick, Dan Gilbert, Leo Slazard, Watson, and Maurice Wilkins, all read Schrodinger's book, and by their own accounting, were converted from working in either physics or chemistry to working in the new area of molecular biology. And if you ask any molecular biologist today and give them any one of these names, they would all claim that these people were molecular biologists, not theoretical physicists. So, marching on to more recent people, Eric Lander, who had a doctorate in algebraic coding theory from Oxford, came to North America. He founded the Broad Institute at MIT and then the Whitehead Center for Genomic Research at MIT, and more recently was a short-lived science advisor for President Biden. He apparently did something inappropriate and had to resign. But again, he played a major role in terms of heading up the Human Genome Project and actually ensuring that we had the first sequencing of the human genome. So there's been an explosive study of nonlinear PDEs, which really started with Hodgkin Huxley's work. Nonlinear dynamics has seen a really massive growth, which was, I think, largely fueled by the discovery of chaotic dynamics. And this just goes on and on. And when we look in the biological areas, we see that physicists and mathematicians are increasingly more interested in biological problems, and they actually understand that that's where some really interesting stuff is to be found. So if we look at the professional organizations, you know, the SciEM Life Sciences group is almost 900 people now. The Society for Math Biology is 800. So there's a huge interest on the part of mathematicians and physicists in the biological sciences. So if you look back at these areas of molecular biology and neurobiology, what made the possible successful collaborations work? Well, I think in excitable cell physiology, the fact was that we had extremely good temporal data. And plus there was an obvious connection between the physics of electricity and magnetism, as well as the biological processes. And in terms of molecular biology, we've also had a huge amount of really wonderful data and infusion of a pool of extremely talented physicists and mathematicians that started after the Second World War. Mathematicians were used to thinking that much of mathematics was being driven by problems in physics, and this persisted up until the end of the 20th century, I think. But it's now pretty clear that mathematicians are realizing that problems are being driven by the biological sciences in this century. And for life scientists, well, first of all, mathematics and physics have played a major role in defining the life sciences, and they're going to continue. It's going to continue at an ever accelerated pace, and people cannot ignore physics and mathematics if they're in the biological sciences, because if you do, you're going to be left behind. You will not be able to compete effectively, because the most effective scientists will be the polymaths, the ones who know the biology, know the physics, and know the mathematics. And if you're really interested in trying to span all of these areas, my advice to you is learn the mathematics and learn the physics first, because actually learning the biology is pretty easy by comparison. So thank you very much. I'd like to thank my good friend and colleague, Moises Santillan of Monterrey, Mexico, because Moises and I started this project a number of years ago, and we had a great deal of fun looking to finding all these connections between the various areas of science that we had not known about. So thank you, and that's it.